It's always a pleasure to be here at the MESA meeting, and this is probably my fourth year, so some exciting developments at Cynata that I'd like to bring you up to date with. First, let me draw your attention to our safe harbour statement, and please do refer to our filings on the ASX, the Australian Stock Exchange website, and also our company website. A brief overview of the company and our achievements in the past 12 months or so. A reminder, we are an Australian Stock Exchange listed company developing an allogeneic mesenchymal stem cell technology utilising induced pluripotent stem cells, or iPSCs, as the starting material. We call this technology Cymerus. It originated from the University of Wisconsin, very much the home of stem cells, and the inventors on our patents include such luminaries in the field as Jamie Thompson and Igor Slukvin. We commenced this year a world-first clinical trial in graft versus host disease. I'll explain more about that in a moment. We are underway with that trial at sites in the United Kingdom and Australia. We also announced this year a strategic partnership with Fujifilm Corporation, who are our 9% shareholder in Cynata. And of course, Fujifilm requires no introduction as one of the most significant players in the regenerative medicine industry. We also have a license option agreement with a German company called Abseth for several disease areas. We have a strong balance sheet with no debt and a cash runway out until 2019 based on our current projections. Our preclinical program has yielded compelling data from a range of animal proof of concept studies attesting to the utility of our MSC product. We also operate, as I'm sure you're all aware, in a favourable regulatory environment, particularly with respect to the potential for fast-tracking incentives. And finally, MSCs demonstrate very broad clinical potential in a wide variety of diseases, including some of the major unmet medical needs that face our ageing populations in developed countries. To draw your attention just to a couple of the elements on this slide, a few key facts. The company's market cap is around $47 million, so we believe at current valuations we are significantly undervalued. And also one of the more significant developments at Cynata in the past 12 months or so has been the appointment of Dr. Paul Wootton as chair of the company. Paul is known to many of you as the former CEO and president of Cynata Therapeutics, which was acquired last year by Astellas Pharma in a $380 million cash transaction. Cynata, as I mentioned, has a major shareholder in Fujifilm with a 9% holding. Company's got cash out till 2019. The company is now very much at an inflection point given our strategic partnership with Fujifilm and the delivery or the execution on our commercial plan, and also commencing a phase 1 clinical trial. As I mentioned, this is a world first, the first clinical trial using an iPSC-derived allogeneic product. Dr. Takahashi's program in Japan, which also initiated this year with an allogeneic product, is a clinical research program as opposed to a clinical trial. So certainly a landmark not only for the company but also for the industry, as it is indeed the first allogeneic iPSC trial that's underway. So what is it that's retarding or arresting the acceleration of the development of MSCs? We believe it's the dilemma associated with the manufacture of MSC products as opposed to their clinical safety or efficacy. The manufacture of the product is holding up the industry significantly, and it could be no better put by Jacques Galipeau, one of the more significant commentators on MSC therapeutic potential, where in his analysis of the failed prochimal phase 3 study, he drew our attention to the fact that it is the excessive culture expansion, as he puts it, being the most egregious divergence between the commercial and academic MSC products as being the scale of product expansion. And that is very much our mantra. We believe that the challenges associated with bone marrow or adipose tissue derivation of MSCs result in a major roadblock for the clinical development of MSC-based therapeutics. And it's summed up on this slide here, where first-generation techniques for manufacture, which rely not only on multiple tissue donors but also massive cell expansion, involves a significant problem. Those challenges are associated with the regulatory issues involving multiple donors. Every donor is different, so therefore every product being derived from each different donor is in turn different, and that's a major regulatory barrier. The recruitment and qualification of donors is a logistic nightmare. Obviously, with intra- and inter-donor variability, we end up with a variable product with inconsistent product quality, and the expansion required to manufacture sufficient quantity from a primary derived MSC results in senescence and reduced product efficacy. This is now well established in the literature. Cynata's Cymerus technology, utilising iPSCs, overcomes each of these challenges. And just in summary here, we try to illustrate the difference between first-generation techniques requiring multiple donors and massive expansion from our technique, which requires one donor, one time, and the utilisation of the massive expansion potential of iPSCs to generate essentially an unlimited starting material of consistent and reproducible quality. The end product, at the end of the day, is an MSC, as according to the ISC classification, but we believe a consistent and more robust product. Our first clinical indication is graft versus host disease, GVHD, which we believe is a logical proof of concept indication. MSCs have already shown significant efficacy, especially if they're freshly isolated MSCs. The trial duration is very short, which of course satisfies the needs of impatient investors for clinical data, and we expect completion of this phase 1 study early in 2018. And a successful outcome of that trial opens the door for our cells in a range of other clinical indications in which MSCs have been shown to be effective. More particularly from a financial perspective, Fujifilm's own projections of their potential market for GVHD are around $300 million at peak, which would result in revenues to Cynata of at least $30 million per year, which of course on a 5 times PE leads to a $150 million US market cap for Cynata. Bear in mind that we're market cap around $47 million today. Some data here to show the efficacy of our MSCs in a GVHD model. This was a murine model of GVHD undertaken at the University of Massachusetts, Amherst, showing in a Kaplan-Meier plot the survival rates of mice who were untreated, that's the blue bar, versus mice who were treated with 2 different doses of our product, showing a statistically significant prolongation of life. The top line there represents animals who received only the cells alone but no GVHD. So this gave us great confidence and obviously was sufficient to enable the approval of our clinical trial application in the UK, which was granted in September of last year. The clinical protocol for our GVHD trial is summarised here in this slide. As I mentioned, it is the world's first allogeneic iPSC clinical trial. The patient population is 16 adults with corticosteroid-resistant acute GVHD, 7 clinical sites in the UK and Australia. Obviously, being a phase 1, the primary endpoint is safety and tolerability, but our secondary endpoints include efficacy markers such as complete and partial or complete response, being the complete absence of GVHD, and partial response being improvement of at least 1 grade, and bear in mind that GVHD gradation is 1 to 4, 4 being the most severe, and overall survival at days 28 and 100. The current status is that the first patient was treated back in May and recruitment is ongoing. We've had subsequent patients since then, and as I mentioned, we expect completion of the trial sometime in the first half of next year, with a DSMB, Data Safety Board review, in around the fourth quarter of this year. It's a little bit hard to be definitive about time frames, given this is a very rare disease and patients don't come along every day of the week. But nonetheless, based on the historic diagnosis rates at the recruiting centres, this timeline is roughly what we expect. From a commercial point of view, the next steps of our relationship with Fujifilm involve Fuji deciding at the end of our phase 1 whether to proceed with exercising the option, which would then result in Fuji being granted a worldwide exclusive license to the GVHD product, and that is accompanied by an upfront payment of $3 million US, and then obviously future royalty, milestone and royalty payments down the track. Importantly, for a small biotech company with limited capital, such as Cynata, Fujifilm will pick up the development costs for the GVHD product post-phase 1, and obviously that will be a huge advantage to us to get the costs off our P&L. Cynata's business model is very firmly focused on partnering our technology. We believe that generating early revenue streams emboldens shareholders who in Australia are generally fairly impatient and don't have the depth of capital and patience that investors here in the United States and even in Europe have. So ensuring that we have the validation that comes with partnering is a very, very important element of our business model. We have a vigorous partner engagement program underway, and our deal with Fujifilm and also with Abseth indicates a very solid execution on that strategy. We externally undertake product development collaborations, and our programs thus far in GVHD, asthma and respiratory disease, heart attack, vascular disease, in particular CLI, and cancer glioblastoma have already begun to yield great results, some of which have appeared in the public literature over the past year or so. We operate in a fairly dynamic market from an M&A perspective, and as I mentioned before, our chairman was most recently with Cynata Therapeutics, which was acquired by Astellas last year. We've seen the spectacular investment by Bayer and Versant in the creation of Blue Rock, who are up straight after me, so stick around to hear what they've got to say. And also, of course, the acquisition by our partner Fujifilm of CDI back in 2015 are elements of the exciting M&A environment that the MSC and stem cell sector is in. So finally, just to summarise, Cynata has the scalable world-first technology that overcomes the inherent challenges that we believe exist in the manufacturing methods for therapeutic MSCs. Our technology is already being validated and monetised through our license agreements with both Fujifilm and Abseth. We have a clear regulatory path. The company met with the FDA in a pre-IND meeting a couple of months ago with a very favourable outcome, and our programs to satisfy the FDA's requirements are underway, and we expect to submit an IND next year. Our clinical trial is underway with data to flow in the DSMB analysis late this year, and then completion of that trial in next year, and some very exciting near-term news flow. I'd like to thank you for your attention, and I see we have about 55 seconds for questions. If there is a burning question, I can probably address it in that quick time. No questions? Well, thank you, ladies and gentlemen, for your attention, and I'd also like to thank the Alliance for Regenerative Medicine for giving Cynata the opportunity to present to you today. Thank you.